Hello, good morning for me, good afternoon for others. I am Fernanda Coelho, and I'm here with Fabiana Canini, who you already heard, giving some information on presentations. We both are the conveners of this session 30, entitled Environmental Factors Driving Diversity and Composition of Fossil and Living Antarctic Communities. We are happy that we could finally make this part of the conference to happen, because we are in a very hard situation worldwide, but I believe we will have good times here. So we will have in the first part of our session, we will have 5 talks, as Fabiana already said to you. Dr. Paulo Câmara is not able to present his talk, so we will anticipate the other 3 presentations. Okay, so I pass the word to Fabiana.
Okay, we can start with the first speaker, who is Professor Jan Eitz from University of Waikato. The title of the presentation is Who Fill the Gaps? Microbial Communities Colonizing Newly Created Aquatic Habitat in the Antarctic Dry Valleys Converge Rapidly with Pre-Existing Assemblages. He is not present due to time zone differences, but we have his presentation recorded, so I will play the video with his presentation. The volume is quite low, but the original video has this low volume, so is the maximum that we can do.
Newly created aquatic habitat in the McMurdo Dry Valley region of Antarctica. The primary contributors are listed here, and a particular shout out to Jose, a student on the project, did most of the actual work. Our location, the Dry Valleys, the largest area of ice-free land in Antarctica, are located here, pretty much due south of New Zealand, and the valley in which we're focusing our attention is the Right Valley, indicated by the blue arrow on the right-hand side of the screen. The reason for our work focuses on the fact that we've known for some time that aquatic habitats are increasing in size in some of the McMurdo Dry Valleys, and we know that this newly created habitat is being colonized, and the question many people are asking is whether this new habitat is creating new opportunities for new species and to enhance species turnover in these community systems. Implications for management, of course, are our desire to protect existing biodiversity in a changing world, and the extent to which we need to take special initiatives to allow that to happen. Let me take you to our study site. This is Lake Vanda, and this is our lab tent out there on the ice of Lake Vanda. But I want you to just draw your attention to the far side of the lake, and just blow that up a little. You can see these bands on the hillsides, which represent historic lake levels. The lake is clearly a dynamic system. It's a closed basin lake, which means that the level is set by the relative amounts of water flowing into the lake, which is primarily driven by glacial melting, and water leaving the lake, which is driven by sublimation and evaporation from the lake surface. So as climate changes, the lake level responds, and it's at the moment we're in a period of lake level rise, which means we're losing soil but we're gaining lake. So the lake level has risen by around about 5 meters in the last 30 years and 15 meters in the last 80 years. And so we've created a significant amount of new habitat during that lake level rise. Our study area incomplete is a hydrologically connected system of lakes and rivers. So we flow from the water source, which is the lower right glacier, melting of the lower right glacier. It first flows into Lake Bramworth, and then the Onyx River carries the water from Lake Bramworth down via the Onyx River to Lake Vanda in the bottom of the valley. We sampled from various parts in this continuum. Methods were to primarily we're focused on microbial mats, which is where we've got the green dots from. So we've collected microbial mats from each of these locations, from Lake Brownworth, top of the system, from the Onyx River, the middle of the system, and from various parts of Lake Vanda. And I want to mostly today talk about the seasonally frozen moat community, which is on the edge of the lake. This is frozen during winter and it's ice-free during summer. It's quite a different environment to further deeper down into the lake, in these green dots further down, where it's under perennial ice cover, but it's also perennially liquid water. So we have a transition from soils through to moat systems, and then from moats through to these perennially wet systems as the lake level rises. We're going to focus on genetic taxonomic data in this presentation, looking at 16S and 18S RNA gene sequence data, and we're going to use those as metrics of prokaryotic and eukaryotic communities respectively. So we're going to be looking at microbial mat community composition. We're going to be comparing it a little bit with some of the water samples which correspond to those microbial mats in the same locations, and we're going to look at how those things change across the environment and infer some changes through time from those observations. Okay, so the transition we're seeing is from dry land, which in the McMurdo Dry Valley looks pretty much like this, we call it dirt, through to a moat system. This is a moat system about 2 meters deep. It's been inundated for maybe 6 years, and it's seasonally frozen, seasonally ice-free. And then finally, much more complex and elaborate microbial mats develop deeper in the lake. This is at 20 meters depth. It's been inundated for maybe 75 years, and this community has evolved over that time period. So it's perennially ice covered, but it's liquid water all year round. So let's first look at our evidence for connectivity amongst these hydraulically connected systems. So the green dots here represent the cluster of points over on the right-hand side of this NMDS plot. So this basically shows the similarity of microbial communities. This is focusing on cyanobacterial communities in this case, the similarity of those to each other in this two-dimensional plot. The closer they are, the more similar they are. And we are seeing a really tight collection here of the Onyx River sites. So these sites down here in the Onyx River, the Lake Brownworth sites, so these sites up here at Lake Brownworth, then the moat sites where the Onyx River enters Lake Vanda. These are these sites down here, and the water that's flowing down the Onyx is all part of this same cluster. So we're looking really at a connectivity of species as we go down this hydrological connection. The orange spot over here are these lakes over here on the left-hand part of the scaling plot, which are on a small island, which is not hydrologically connected with the green dots, and we're seeing that we're having a degree of separation of the communities on that location from those that are being directly affected by the Onyx River site. So this is our first evidence of connectivity. So as new habitat is created, it's being initially potentially affected by the source of propagules available to it. So let's look at what happens as we go through a kind of a succession process within a moat system. Here we're looking at sites we're looking at 2 replicates from each of 4 moat locations at Lake Vanda, and we're looking at them from this range of depths over here. That range of depths we can correspond to the number of years that the water has been covering the soils there. So again, from 1 year through to around about 6 years as we go from the edge of the lake down to around about 2 meters deep. And what we can really quite clearly see here is that for prokaryotes, in this case, it's all bacteria. We've seen this succession process where we very quickly move from, we select very quickly and a new, we have a succession process going on from 1 year through 2 years and 3 years, 4 years, 5 years and 6 years until we're ending up in this area here in the 4 to 6 year period of a tight cluster of communities which are very similar to each other and in fact not significantly different. The same is true for eukaryotes. Here again we've got the same samples, but we're looking at eukaryotes. We're looking at the change from 1 year, very quick selection process through to 2 years, and then we end up with this very similar gradual succession process through to by the time we get 4, 5, 6 years old, the communities are indistinguishable from each other. So the second process we're looking at here, we looked at colonization initially. We saw there was a degree of response to connectedness, but we're now looking at how the community evolves as it develops in the moat system, and we can see this convergence across all of these sites, which are separated from each other by hundreds of meters, and we're seeing this convergence towards a standard moat community after 4 years. In this plot, we're looking at the shallow water sites, which is the pale blue, this section over here, and we're also looking at the deep water locations in Lake Vanda. So I mentioned earlier that these shallow sites, they all have a relatively similar environment in that they are frozen during winter, liquid during summer, and these, we're looking over here on the right hand side at communities which are from deeper in the lake, from 8 meters plus, which are under ice cover all the time, but they are also liquid all the time. So we see a very strong separation. If we think of these 6 points here are our moat communities, so these the moat communities within the lake, and if there is a transition going on as they transition from being moat communities, as lake level rises, they're going to transition to being under ice moats. They're heading in this general direction if we're going to sustain the same type of biodiversity that we see there. If we look at this cluster of deeper mat communities, then we can see that they don't separate very well on the first axis of the NMDS, which really separates the shallow water from the deep water communities. But most of the separation is by NMDS. It's the second axis of the multidimensional scaling array. If we plot axis 2 score against depth, what we can see is, again, we can see a well-organized pattern here where we have a succession of community composition as we go through down the lake, which is we can look either as a long duration succession, or we can look as a response to the changing environment as we go deeper, lower irradiance in particular. But there's a high level of similarity between organisms that have been 100 years, 75 years, and many more than 100 years below the ice cover. So convergence again here in terms of the NMDS scores of communities at great depth, and this clear structural change which we can potentially attribute to either a habitat characteristic or to a successional process. A consistently successional process which appears to be driven by habitat. So how is new habitat being occupied in these systems? So it's no doubt that on initial inundation, and the colonists are from the soils, but they're also from inflowing water, and they're from local communities, because we're seeing this connectivity effect and this similarity across the environment. Over relatively few years, maybe 4 to 5 years, and then the successional processes within the moat system, the newly created moat environment, they drive the community towards what we can call a standard moat community. And we infer that this is driven by the environmental conditions. So there is environmental filtering which is determining the transition through to this standard moat community. We have the existing moat community, which no doubt is potentially able to provide resident organisms to carry on colonization of the moat community as it slowly increases. It's increasing at around about 30 centimeters a year, so it's not a huge demand for movement for transfer of organisms. And then our secondary transition, as we go from moat to lake, and we go to increasing depth in the lake, we're also seeing this organized situation where we're getting a predictable communities occurring in predictable habitats. So our inference right now is that, while colonization does create that potential for turnover, then that selection by habitat is sufficiently powerful to sustain our status quo of community composition over time. So while these habitats are getting bigger and exposing new habitat to being colonized by microbial communities, what we're actually seeing is that because the habitat is tending to remain the same, then habitat selection is sufficiently powerful for us to retain the key elements of our biodiversity fingerprint for each of these habitats. So I'm going to stop there. Thanks for your attention. As usual, these are our supporters, financial and logistic, and we I hope we get a chance to talk about this at some state in the future.
Okay. So the audio was a little bit low, but I think it was possible to hear the video by Ian Hallways, and we will have opportunity to discuss all presentations next week. So we go for the next one. The talk is by Becky Ball, and she also sent us her video because of time zone issues. So Fabiana will play the video. The title is Habitat Severity Structure Soil Communities and Above Ground, Below Ground Linkages Across a Latitudinal Gradient on the Antarctic Peninsula. Please, Fabi.
Hi, my name is Becky Ball, and I'm going to talk about some of our work looking at the impacts of plant type on soil communities across a span of over 2,000 kilometers on the Antarctic Peninsula. The peninsula is experiencing rapid environmental change. Rapid warming over the late 20th century is predicted to resume and continue over the rest of the 21st century. That changing climate makes the peninsula susceptible to changing biotic communities. Warming temperatures allows the expansion of native species into new habitats, as well as the establishment of invasive species both above and below ground. The peninsula already has documented invasions of an annual bluegrass, as well as invasive chironomid midge and several species of springtail. Changing plant communities will certainly have below ground impacts. While it is known that Antarctic soils under certain vegetation differs from one another in many characteristics, the exact nature of the influence that different above ground communities have on soil communities is not well described, particularly not beyond the site level. We also know less about the peninsula's soil communities than other ecosystems globally. Much of Antarctica's terrestrial biodiversity is microscopic and has not extensively been surveyed. Our knowledge of the local biological diversity is increasing, but at limited rates due to logistical constraints and harsh conditions. Except for one other project that we're aware of, it's been difficult to compare sites and patterns larger scale. So we designed a field campaign to provide in-depth assessments of biological diversity for a broad range of taxonomic groups across an environmental gradient along the peninsula. We want to know how environmental severity along the latitudinal gradient of the Antarctic Peninsula influences the relationship between plant type and soil communities. One hypothesis is that connections with plants increase as sites become more severe, perhaps because it becomes increasingly important to have a particular microhabitat for protection. But alternatively, it could matter less because maybe it's so harsh and unpleasant that the only survivors could survive anything regardless of their plant type. In addition to that ecological question, we also wanted to understand who lives where and why. The goal being to identify areas to protect without having to put a shovel in the ground. So we sampled from 10 points, a total of 13 sites along 2,300 kilometers of the Antarctic Peninsula, from a southernmost point at Sky Blue on the continent, up along the west side of the peninsula to the southern end of the Scotia Arc, and that includes some previously well-studied sites as well as some very understudied or new sites. At each site, we sampled soil from beneath 5 different cover types. We sampled beneath algae, which was usually Prassiola, moss of various species that dominate at particular sites, Antarctic hairgrass, lichen growing on usually a mix of dead organics, and bare soil as kind of a control. At each site, we took 5 soil samples per cover type, but at 5 of the sites, we did what we call high-intensity sampling, where we actually took 20 samples from beneath moss, grass, and bare soil to be able to do more in-depth statistical analyses. So we collected about 500 samples, and on each of those samples, we measured a comprehensive set of biotic parameters across taxa. So we looked at the microbial community, we did sequencing for bacteria, archaea, and fungi, metagenomics for functional identification, as well as the biomass of those 2 groups using epifluorescent microscopy. We looked at the invertebrate community, so nematodes, rotifers, and tardigrades. We sampled using kind of a modified Behrman funnel technique that was stable for life on a research vessel. And the Collembola mites and midges, we sampled using a similarly modified Tulgren extractor. In addition to measuring the biotic community, we also looked at numerous soil chemical and physical parameters that would relate to biology, everything from moisture to texture, carbon, nutrient content, all the cations and anions of typical importance, pH, salinity of the soil, so a variety of soil and chemical parameters, and we also downloaded climate data for all of these sites. So we have a lot of data. We don't have time to go over all of the details, so this will be a very brief overview of kind of the highlights of what our data have to tell us. Unsurprisingly, soils differed in chemical and physical properties across the transect. The most general patterns were differences between the mineral soils in the southernmost continental sites compared to the maritime sites across the rest of the transect that have much higher organic content. These continental sites had the highest mean pH, so they're more alkaline than the maritime soils, higher calcium content, and also consistently lower soil moisture, organic content, and nutrient content. In comparison, the mid to northern regions are in the maritime ecosystem category with a much gentler climate that allows for more complex biotic communities. These soils are much higher in organic matter and nutrients. Interestingly, the mid-latitude sites around 67.5 degrees south stood out from the other maritime sites having the lowest average pH and consistently among the highest soil moisture, nutrients like nitrogen, phosphorus, and potassium compared to sites to both the north and the south. That is pretty much where latitudinal patterns end. There were a lot of site differences in all of these properties that were not explained by latitude or any overarching climate characteristic. An NMDS of a subset of soil and climate parameters that were identified to be non-collinear with each other demonstrate how sites varied along a gradient of overall habitat severity, ranging from more favorable conditions of warmer temperatures with moderate precipitation and high nutrient but low pH soils in the upper left quadrant, to sites that were either wetter or drier, often cooler, and notably low in nutrients. Notably, this measure of habitat severity did not change linearly with latitude because it's mid-latitude sites that are the least severe, followed by a mix of some of the high and mid-severity sites. Another interesting point is that Mars and Aries oases, which are continental sites, group with the other maritime sites, though at the harsher end of the maritime sites. So keep this spread in mind as we look at the biotic community. We'll start with the microbes. For alpha diversity, here we see expected diversity Chow-1 of both bacteria and fungi according to latitude. So the sites are listed from northernmost to southernmost. And we see that fungal diversity is roughly related to latitude. Bacterial diversity, however, didn't change according to latitude. However, if we rearrange these sites to be in order of habitat severity, according to where they fell on the NMDS, we do see a relationship. So we see that bacterial diversity is in general much higher at the low severity sites and decreases as they become more severe, with the exception of just one site. Thinking about beta diversity of both bacteria and fungi, total beta diversity, so Sorensen dissimilarity, significantly increased with distance along both the habitat severity gradient and latitudinal distance. So in other words, bacterial communities are increasingly distinct from each other as the physical distance among the sites increases and their difference in habitat severity increases. And this pattern corresponded to increasing turnover, suggesting that the change in beta diversity with habitat severity and distance is the result of species replacements. So those are some interesting but brief patterns across the peninsula. What about that relationship with plant community? Bacterial communities differ according to the plant community that's growing above them. But what's most interesting is the relationship to plant communities as habitat severity increases. These are results of an NMDS of the bacterial community composition depicted according to cover type, and the sites are listed from least severe to most severe. At the low severity sites, communities under different cover types tend to overlap. As severity increases, they start to differentiate a bit, though not enough for an analysis of similarities to detect a significant difference, even though a Permanova did find differences among cover types at these sites. It's at the mid-severity sites where we see a statistical difference among plant communities. This then disappears at the most severe continental sites. Results for the fungal community were the same, though in these data we only have fungal community data for a subset of the high intensity sites. But again, it's at the mid-severity sites where cover type influences fungal community composition. Invertebrates, however, responded differently. Here's what's important for invertebrates. Again, abundance did not vary linearly with latitude for any of the taxa. Interestingly, those site differences also did not correspond to habitat severity the way microbial communities did. So when we rearrange the sites according to least to most severe, we don't tend to see strong relationships. Springtails are really the only taxa that somewhat closely align with that habitat severity gradient, but there's of course a lot of sites that are exceptions to that. The different responses of microbes and arthropods to habitat severity reflects the different habitat characteristics to which they correlate. So while bacterial diversity was correlated with most of the climate factors included in the NMDS of habitat severity, arthropod taxa are related to fewer of them. Springtail abundance, like bacterial diversity, was positively associated with temperature variability, suggesting that they are more robust against cold temperatures and comparatively less sensitive to precipitation. But in comparison, mites are less robust to temperature variability and cold temperatures and prefer consistently wetter conditions. But it's not just about climate, also soil habitat. Bacterial diversity and springtail abundance again responded similarly, being positively correlated with measures of organic matter content like loss on ignition and total percent carbon, nitrogen content, as well as other macro and micronutrient elements. Mite abundance was most strongly correlated with aluminum content, with weaker correlations with other elements, and rather robust to carbon and nitrogen. How does that above ground plant type matter? Overall, Across the invertebrate taxa, they tend to prefer grass and decline in bare soils, but they differ in their relationships with other cover types. So that was our very quick overview of some of the most interesting points in our data. Just to summarize, we found that soil communities respond to a complex set of soil and climate parameters that we summarize as habitat severity. And above ground, below ground connections appear to be strongest not at the high or low severity sites, but at mid severity sites. We also found that beta diversity is likely caused by dispersal limitation and other historical effects from species turnover or species replacements, not environmentally deterministic patterns. So it does not appear to be the case that the stress-tolerant generalists live everywhere and only the less robust drop out as habitat severity increases. Therefore, conservation efforts can't focus on just high biodiversity sites because it's not capturing all of the biodiversity. And we also show that the relationships vary among taxa, which tells us that if we want to preserve biodiversity, we can't rely on a single or a small handful of habitat characteristics as a catch-all for all soil biodiversity because they respond to different climate and soil habitat parameters. So thank you very much for listening to my video. My contact information is here on the left side of the slide, including a QR code that you can snap just to go right to my website with my contact information. And I'll just end by thanking the small army of students that helped us process all 500 of these samples, as well as our funding from the National Science Foundation and our field and logistics support from a collaboration of the U.S. Antarctic Program and the British Antarctic Survey.
So we are going to move to the next speakers. A live presentation by Mrs. Christina Davis of University of Florida. The title of the presentation is Composition and Diversity of Microbial Communities in Subglacial Lakes Beneath Seapole Coast Ice Streams. So, when you want,
it's saying I have it's not allowing me to share. Let's see. Oh, okay. All right, let me try this again. Can you see it now?
yes, it is loading. We can see it now.
Oh,
so.
Good to go?
Yes, when you prefer, you can start.
Hi, I'm Christina. I'm part of the Brent Christner Lab at the University of Florida, and I'll be talking about the composition and diversity of microbial communities in subglacial lakes beneath Seapole Coast ice streams. So here is a picture of the borehole we drilled a kilometer through the ice to access a subglacial lake. And so this project that I was on is based off another project called Wizard that sampled 2 other subglacial sites. So one was a subglacial lake, Willans, which you can see here. And this was then followed by a grounding zone sampling event that's done at the terminus of where the water was flowing out of this Willans Lake. So we learned a lot from this first project that then set the stage for the Salsa project, which was sampling Mercer Subglacial Lake farther south. So from what we learned from the Willans is that there are microbes that exist in these dark ecosystems beneath kilometer of ice. These lakes are also quite diverse in the microbes that we find there. And so this drove our primary hypothesis that subice hydrology and relic deposits of marine organic matter regulate the ecosystem processes in these active subglacial lakes. And so we kind of drew a schematic. So on the left, you can see our diagram of the possible microbe nutrient cycling pathways of how we think carbon is moving through this system. So with these lakes, they are hydraulically active. So we have an inflow of water that could be feeding various organic matter, but also microbes could be coming and then they colonize these lake units. There is also some evidence that there were past marine incursions, with the most recent being about 10 to 15,000 years ago. So with that, that would deposit a whole bunch of organic matter that would be coming from the ocean. And so this research is very important because it's helping us understand how carbon is cycled, and this water is also moving out into the ocean ecosystem. So if we have a lot of methane or carbon dioxide that's building up in these systems and flowing out into the ocean, it can cause ocean acidification, which also isn't good. So if we can get better estimates on how much is moving out of the system, it'll help for better predictions with climate change models. Also, by understanding these nutrient cycling pathways, it's helping us kind of learn and understand what it takes to survive in an extreme environment in the cold and in the dark that may also resemble life on other ocean worlds. So as I said earlier, that these lakes are hydraulically active. On the right, you can see a hydraulic map of where the water is sourced from, at least for the Mercer subglacial lake. The Willans has a bit different area of where it's gathering its water in the catchment. But these lakes also have fill and drain cycles. So this was discovered about 20 years ago, that subglacial lakes exist, and it's based off of GPS and satellite imagery that noticed changes in the rise and fall of the ice sheet. So when the ice sheet was rising, these lakes were filling with water, causing a buildup and pushing that ice upward. But as soon as they start draining, you can see that that ice sheet then begins to go down. And so this cycling process kind of depends on the lake, but it could be impactful to the microbial community that we're seeing in each of these lakes. So for accessing the lakes, we need to do this in a very clean fashion. So we used a clean access hot water drill. I won't get into it, but if you want to learn more about it, I suggest checking out Priscu et al. From 2013 and Mouchad et al. From 2020 on the methods that we used, as well as some of the results of how we know it's a clean system. So moving into some of the first measurements that we were able to gather from Willans and Mercer, the lakes are pretty similar in what we're finding. Mercer was under a kilometer of ice, and the water column depths, how I was talking about, it's a hydraulically active. When we sampled Mercer, it was just starting its drain cycle. So we measured that water column to be about 15 meters deep. But when they sampled Lake Willans, it was near its finished draining cycle and even starting to fill back up. And it was about 2.2 meters height for that. The important, potentially for the microbes that we measured, were the dissolved oxygen concentrations and the dissolved organic carbon concentrations between the two lakes. So Willans, they measured lower oxygen concentrations, but much higher dissolved organic carbon. And with this, you can see in the cell densities that they are much higher in the Willen system than what we were seeing in the Mercer system, which has much lower dissolved organic carbon as well as much higher oxygen concentrations. And this is also supported with the doubling times of the organisms we found being about 4 times longer in Mercer to have a generation time. Some unique features that we saw in the sediment cores are, if you look at this right image, this is a multi-core. It looks pretty similar throughout. The top looks a little bit more watery, but it wasn't until we did CT imaging of the cores that we noticed distinct features about them. So the top 10 centimeters, they were a bit soupier, but we see that there's a lamination happening, which could have been from different fill drain cycles, and we're not quite having the ice completely touch down during that draining, which could be good for the microbes. Then they're not being forced out of a habitat or smeared or moved out of with the glacier movement. But it also, since it is very porous, it's a good space for these microbes to acquire nutrients and water for cycling pathways. Also of note, about 30 centimeters down in the core, we saw that it becomes an anoxic zone. So this is definitely going to cause community shifts. And it becomes more compacted as well as we move through the core. And we were able to get a gravity core about 2 meters in length, but we pretty much look the same as the lower diamic compacted region, but we did see a strange sedimentation band about 65 centimeters down in it. And so our thinking maybe this could potentially be a marine incursion that deposited a whole bunch of clays and nutrients, which that could impact the microbes that we're seeing in this region, either having some represent some ancient marine organisms, or it's depositing a layer of organic matter that may be rich for organisms to feed off of. We're still looking into this, but definitely a feature that we saw that could be impactful. So then we measured primary and heterotrophic production within the lake water columns using carbon-14 bicarbonate and hydrogen-3 leucine. So these were incubated with this, the cells to determine primary and heterotrophic production of carbon within the water samples. And so the SLW, they saw that there was a positive net production of carbon. So this could help, going back to what we were seeing with the limnological parameters, that it is a more productive lake, that we are getting more generation of that carbon that could then support a heterotrophic community. However, with Mercer, we see that there's a lot less production within the lake, but it's also negative. So it's the heterotrophs are more productive in cycling the carbon than we're seeing with autotrophic production in these environments. And we can't really consider the grounding zone data because there was high variability between the replicates. So moving into an NMDS ordination plot using 16S ribosomal RNA genes data, we're looking at samples taken across all 3 sites with clean access sediment and water. That samples that were taken. So the sediment samples, we have multi-core and gravity core only for the Mercer samples, but the water, it's size fractionated, going from 10 microns in diameter down to 0.2 microns in diameter. And so with this, we see a trend where the sediment seems to be grouping all together in this middle region here. And then the water columns are quite separate. So going back to the hydrology of the system, this could be that the source waters for these lakes are quite different. And so with that, they're providing a unique microbial community in those filling and draining cycles. The sediments, however, they don't move around very much, and maybe they're more impacted by a larger climatic event like a marine incursion, which is why we're seeing them kind of grouping closer together. It may help stabilize or hold the nutrients with the communities that inhabit them. But also, when we look at the, including the gravity cores, we see that the surface sediments from the multi-cores most closely resemble that of the Whillans Lake as well. But as we move farther down in the column to the gravity cores, which are about 2 meters down, we see that it shifts over to where the grounding zone samples are. So again, this is kind of providing support that these sediments may be influenced by a marine event. And when we look at the community and what's shared between all of them, there's not many shared across all 3 sites, as we would expect, with the grounding zone being marine and the 2 lakes being a freshwater system. But within the sediment, we see several ASVs that are shared between the 2 lakes, 667, and that the Mercer sediments are very rich compared to what they found in the Whillans. This could just be that we have a lot more samples, and also going further down depth-wise within the cores. But when we look at the water column, there isn't as many shared taxa, and the water column is more rich in the Whillans than in the Mercer. So by looking at this, it's kind of helping us establish what may be a core community of the sediments and water column for a subglacial environment. However, by looking at some of these unique taxa within Mercer and Whillans, it may help us establish and understand what may be driving these taxonomic changes and community changes within these subglacial lakes. So with this, we see that there are many sources of organic matter and microbial input that could be influencing the community that we're seeing in these subglacial lake systems. So we have flow from water up glacier, potential past marine incursions, basal fallout, so the ice is melting and accreting that's over these lake areas. And we also, deep within the sediments, there's a community of, I didn't get into it, but methanogens that are producing methane, which seep up, and we have evidence for methanotrophs who are feeding off of the methane and putting carbon into usable form for the heterotrophs. And so when we look at these different parts and try and understand what they're influenced by, it's seeming that the water column may be more influenced by these upglacial water flows, while the deep sediments may be more impacted by a past marine incursion. This is still to be completely answered, but with future work, we're hoping to determine what are the nutrient cycle pathways that are occurring and who are the organisms that are primarily responsible for it, and also understanding what's driving the community composition changes so that we can better understand these subglacial environments. And so I'd love to thank my team and NSF, and if you want to follow our project, it's at Salsa Antarctica.
Okay. Thank you for your presentation. The next one is Peter Leroux. I think the pronunciation is okay. Examining the complexity within a key plant interaction. Interspecific facilitation mediates the outcome of intraspecific integrations in the subantarctic.
Right, thanks so much. Yes, I've got a video that I'm just going to share. I was a bit concerned about our connectivity. Let's just maximize this. There we go.
Hi everyone. Thank you for being here online to join this presentation. My name is Peter Leroux and I'm presenting work done by a former PhD student of mine, Morgan Rath-Creer, in collaboration with Christian Schorp and Melody McGeoch. I'd encourage you to also have a look at Morgan's virtual display, which is based on a different but related component of her PhD. So in this presentation, I'll be telling you about how we examined both the inter- and the intra-specific interactions between the two most widespread vascular plant species on subantarctic Marion Island. The outcome of interactions between plant species can vary spatially and temporally, and often depends strongly on the identity of interacting species. However, generally speaking, interspecific interactions are assumed to be negative due to competition. Strong competition is also expected between individuals of the same species. For example, when individuals of the same species are grown in high-density stands, individuals from low-density stands may perform better than individuals from the high-density stands. Therefore, both inter- and intraspecific interactions may strongly affect plant species performance, with negative outcomes being expected and often being observed. However, in stressful environments, we often see positive interactions between species too. The stress gradient hypothesis posits that these facilitated interactions will be more frequent and more intense under increasingly severe conditions. And indeed, the majority of studies support this idea. Therefore, in stressful environments like Antarctica and the subantarctic, we expect at least some interactions between plant species to be positive. A common example of positive interactions in severe environments is facilitation of other plant species by cushion plants. Cushion plants are low-growing compact species that are often long-lived and typically increase resource availability and ameliorate abiotic stresses locally. For example, several cushion plant species are known to improve substrate stability, as well as increasing the availability of water and soil nutrients, while also buffering substrate temperatures. As a result of these impacts on their local abiotic conditions, it's frequently seen that cushion plants have positive impacts on associated plants, including increasing plant species richness and vegetation cover. However, it's important to also understand how these positive effects associated with cushion plants affect interactions between the other species that co-occur with them. Okay, let's sketch out some possibilities here. In this diagram, these light green figures represent cushion plants, which are termed benefactor species here, and the other individuals growing on them represent other species, which we will call beneficiary individuals. Within the context of facilitation, interspecific interactions between cushion plants and beneficiaries have the potential to mediate the outcome of intraspecific interactions among beneficiaries. For example, where facilitation increases the size or density of beneficiary species, it is possible that the negative influence of competition between beneficiary individuals could outweigh the positive influence of intraspecific facilitation. In other words, it's possible that facilitation by a benefactor species can result in high densities of beneficiaries, but due to crowding, beneficiaries may not be able to reach reproductive size. Therefore, it's possible that the facilitative positive effect of a benefactor on a beneficiary species could be negated by the negative effect of high beneficiary density on beneficiary reproduction. However, it is also possible that beneficiary species could benefit from higher densities. For example, they may benefit by sheltering each other from strong winds or by increasing substrate stability. Therefore, the aim of this study was to test whether interspecific facilitation between a benefactor species and a beneficiary species mediates the outcome of intraspecific interactions within that beneficiary species. Our study is unique because while other studies have documented the outcome of interspecific interactions among beneficiaries within the context of facilitation, in our study system, there's only a single beneficiary species that is common. And that therefore allowed us to explicitly focus on intraspecific interactions without having to disentangle the influence of interspecific interactions between beneficiaries. Our study was conducted on subantarctic Marion Island. Our benefactor species was the dominant and widespread cushion plant species Azorella selago, and our beneficiary species was Agrostis magellanica, a dominant perennial grass species of Marion Island. Azorella selago is known to facilitate Agrostis by, for example, increasing Agrostis abundance and cover. We used both an experimental and an observational approach to test our hypotheses. First, using an observational approach, we sampled Agrostis growing on Azorella and growing on the adjacent substrate along 6 altitudinal transects. The identity and cover of other vascular plant species was recorded, as was the cover of bryophytes. All Agrostis individuals in those samples were harvested and dried for 48 hours before being weighed and measured. Several measures of Agrostis performance were recorded, but the variables that we're going to focus on now is first the proportion of reproductive grass individuals. So that tells us about how successful each individual grass was. However, because examining the total number of reproductive individuals per cushion plant or per sample provides insights into population level reproductive output, we'll also report some results for that metric. We used generalized linear mixed effect models to model the proportion of reproductive Agrostis individuals as a function of whether the grasses were located on a cushion plant or on the adjacent substrate. And that obviously then tells us whether the interspecific interaction has an effect. We also looked at the effect of Agrostis density, which obviously tells us about intraspecific interactions, as well as the effect of altitude, which is a proxy for environmental severity. We also then included 3 pairwise interactions between these variables, since these predictors may mediate each other's effect on Agrostis performance. We used the same analytical approach to also model the total number of reproductive individuals, just adding an additional term to account for the area of each sample. The results from our observational component were striking. First, we observed that the proportion of reproductive Agrostis individuals declined strongly with conspecific density, with this decrease in grass performance being greater on the cushion plant than on the adjacent substrate, suggesting that the facilitative effect of Azorella changes the intensity of intraspecific interactions between Agrostis individuals. In contrast, when we look at the total number of reproductive Agrostis individuals, instead of just the proportion of individuals that are reproductive, we see the opposite result, with sample level reproductive output increasing with density, showing that despite a lower proportion of grasses being reproductive, the total reproductive output still increases with grass density. Second, going back to looking again at the proportion of individuals that are reproductive, we see that the proportion of reproductive grasses was consistently higher at lower conspecific densities, shown here in blue, compared to medium and high conspecific densities across the altitudinal range. Therefore, from this observational data, it appears that intraspecific interactions between grasses are negative, and with this having a particularly strong negative effect in the presence of the positive interspecific interaction. Nonetheless, the net effect of the interaction between these two species is still an increase in the total number of reproductive individuals. Now, moving on to the experimental component. Here we experimentally thinned Agrostis to lower its density within plots, and then compared the performance of Agrostis in these thinned plots to the performance of the grass in high-density Agrostis plots and low-density Agrostis plots. 3 grass individuals within each plot were randomly selected and were monitored for a growing season to determine their survival, growth, and reproductive effort. Since only a few grasses didn't survive the season, and since only a few were reproductive, we'll report here the results for shoot mass instead. These data were again analyzed using a generalized linear mixed effect model, with the influence of Azorella representing the effect of interspecific interactions, and the influence of the density treatment representing the effect of intraspecific interactions. In contrast to our observational component, in this experiment we did not observe any effect of agrostis density on conspecific performance. There was still a significant positive impact of the interspecific interaction, with larger grasses growing on the edge of Azorella selaga cushions and elsewhere. So what do these results tell us? The first key finding from this study is then, when examining the impact of agrostis density on agrostis performance using an observational approach, we see evidence for intraspecific competition. Probably more interestingly, our results suggest that competition among beneficiaries may be mediated by the interaction with the benefactor, as our observational data shows the facilitative effect of Azorella changes the intensity of intraspecific interactions between agrostis individuals, because the decrease in agrostis performance with density was greater on Azorella than on the adjacent substrate. However, this finding was not supported by the experimental component of the study, as reducing agrostis density had no significant effect on agrostis performance, suggesting neither facilitative nor competitive interactions within the grass species. This difference between the experimental and observational study may be due to monitoring only being conducted for one season after the experimental reduction of agrostis density. And this potentially reflects that the grass species may respond slowly to changes in this biotic interaction. Our second key finding was that for both components of the study, the proportional and the total number of reproductive agrostis individuals was on average higher on Azorella selaga cushion plants than in the adjacent substrate across the entire altitudinal range of the grass species. Therefore, in agreement with previous findings, the interspecific Azorella agrostis interaction positively affected agrostis performance, both at the level of individuals as well as at the level of samples. Our third key finding then was that despite the negative effect of intraspecific density on the proportion of reproductive agrostis individuals, the total reproductive effort of agrostis was significantly greater on Azorella than on the adjacent substrate. Therefore, even though high intraspecific agrostis densities due to interspecific facilitation resulted in a lower proportion of individuals being reproductive at high densities, the total reproductive effort of agrostis was still greater when agrostis was facilitated by Azorella. So, more generally, our results suggest that both positive inter- and intraspecific biotic interactions may be crucial for the performance and the survival of plant species in extreme environments. Additionally, where possible, experimental approaches should be used in conjunction with observational data to accurately examine context dependency and the outcome of these sorts of interactions. Within the context of facilitation, the overall performance of beneficiary species may depend then on the combined effect of the benefactor species and the beneficiaries, on benefactor-mediated interactions, and on the interaction of these with their environment. Therefore, to accurately forecast the impacts of environmental change on species, on communities, and even on ecosystems, especially in our planet's most extreme environments, we need to continue to develop our understanding of the influence that biotic interactions have both within and between species. Thank you so much for joining this presentation. To wrap up, I'd like to acknowledge the contributions from a large team that made this research possible, as well as the funding and logistics support of the South African National Antarctic Program through our National Research Foundation. I look forward to hearing your comments and questions on this work. Thank you.
Thank you, Peter. Next speaker is Dr. Annick Cournot from the University of Liège with her presentation on fostering bacterial biodiversity in collaboration.
Hello, do you see my presentation?
Yes, we can see it.
And it's okay, because I don't see it.
Do you want some help? To visualize your presentation.
I see just a screen you're presenting to everyone, but I don't see. So maybe I do it again.
You must choose the pin in the presentation.
Yeah. Yeah. I see a screen you're presenting to everyone, but I don't see my slide.
Now you have to go to the file of your slide and use it as a normal presentation. So select the full screen mode and move it as you see it on your screen.
Okay, I will try. So thanks very much to SCAR and the session conveners for giving us the possibility to present our work. This work was carried out with 3 previous collaborators of the lab, Igor Spessy, David Velasquez, and Dale Laughinghouse. We aim to answer 2 questions. Can herbaria specimen from the Smithsonian Institution give useful information on past cyanobacterial diversity in polar regions? And how should we preserve herbaria samples to give a useful picture of the modern cyanobacterial biodiversity in 100 years from now? So we know that herbarium specimens are valuable, and it's now possible to extract their DNA and so to sequence their biodiversity, even if there are still technical challenges. In 96, Palinska and colleagues have shown that it was possible to extract.
We can see your presentation moving on. It's only the first slide of your presentation.
You can see what? Sorry?
We can the other slide of your presentation. We see only the first one.
Zut. Escape. Yeah, you have a suggestion?
Yeah. Use the full screen mode.
Yes, I use the full screen.
Okay.
So, yeah. So should I use another of the 2 options? Because there are 2 options. When I see present now, your entire screen and a window. And you told me that both were okay, but...
Yes, it should be the same using both the options.
If I open a window, they ask me which screen or window to share. And so I enter my PowerPoint file, and then I authorize the Google to share it.
Okay, we can see your PowerPoint.
Yeah, but now I go to the second.
Yes, we can see it now.
You say okay. Let's hope. So I'm going to that slide where I was busy. So you see it, and yes. So yes, it was possible to extract DNA from herbarium samples, and recently Anne Jungblut and her colleagues have shown that it was indeed possible to extract DNA and sequences from 100 years old Antarctic samples, and also cyanotoxins. Our samples come from Smithsonian Institution, and more particularly from the Francis Drouet Herbarium. So he has devoted his life to the taxonomy of blue-green algae, as they were known at the time, and he legated his herbarium to Smithsonian Institution. And this herbarium was also fed with samples from American expeditions like the Navy surveys or the geological surveys. And Francis Drouet has published about the conservation and preservation of herbarium specimens, and he would advise to use slow air drying. And we will see that indeed it's quite good. In order to win time, I don't give the methodological details, but they will be in the PDF files that I will put on the website. And I can tell you that the DNA was extracted with the power biofilm of Mobio kit, and with all precautions and controls. And the 454 sequencing was carried out on amplicon, which consisted of the V3, V4 region of the 16S ribosomal RNA gene, which was amplified with cyano-specific primers. There were 7 samples, one from mountains in Tyrol, which was quite old, Alaska, 1 sample, and 5 samples from Antarctica. The number of reads was quite different, going from 67 for sample 17 to 16,000 for sample 24. And at the end, 86 OTUs were defined at the 97.
I'm sorry to interrupt you. I'm sorry. You must choose the presentation format of your PowerPoint, because it's set on one slide, third slide of your presentation, because it's not in the presentation mode. It has nothing to do with the Meet. It has something to do with the PowerPoint.
Yeah. So if I click on diaporama, it's supposed to be full screen, and it's not what you see. Huh?
Choose the presentation mode on your PowerPoint. I think it's not sure.
That's what I do. But that's what I
in the foot bar. Oh, right, you're right. Yes, this one.
Sorry, I don't understand. Where are you?
Yes, and also yes, someone here gave some tip. When you choose the presentation in the Google Meet, you must choose to share the whole screen.
It's not asking me that. Present now. So, entire screen. I select the PowerPoint.
No. You choose present now, and then share the whole screen.
That's what I did.
The PowerPoint. You choose the whole screen, and then everything that appears in your computer will appear for us.
Annick, you have to turn on your microphone. We can't hear you.
Yes. So I did all what you wanted, but I don't. Yeah. I am in the PowerPoint. Yes, because if I am in the Google Meet, I don't see what I present. So I go to the PowerPoint. Yes. And then I choose to have full screen, so what we call diaporama, and it doesn't work.
And it should have working. I don't know. Igor Pesci said that ask you if you have two screens. Do you have two monitors?
I had looked at the connection. Yeah, I don't. Yes. Is it better now?
Well, it's the same. Try to change your slide.
Change my slide. Autoriser. Yeah. Painting to the video. Cool. Yes. So is it, do you see my slide?
Yes, we see, but we don't see the presentation mode.
The presentation what?
The mode.
Okay.
When you change your slides, we just don't see it. We see the PowerPoint screen. And one alternative is to change your slide without presentation mode. And you must change it manually on the right bar of your slides. And maybe we'll see the correct slide you want to approach. It's a try.
So you mean that you see on
the that you have the presentation, maybe you can share it if you agree, Anika.
Sorry, I don't understand.
Igor is writing that he has your presentation.
Okay. Yes.
You can share it if you agree.
Okay. No. So I have to stop presenting. Sorry for the problem, but I just don't know what's happening. Yeah, so
now it's perfect.
yeah. Okay, so we had 68 OTUs defined at that 97.5 level of 16S similarity. So next slide, please. So the first sample we are going to look at, so we look at the samples at the diversity and the collector and the habitat. So in this case, it was a sample from a mountain in Austria, in Tyrol. And on the label, it was written some additional information that was not on the website, namely that it was taken at 1,700 meters of altitude in a marsh. So next. So thanks to that name of the mountain, it was possible to see where the mountain was. And next. The sequencing gave us a large majority of one OTU, OTU 3. And next, you will see that it's exactly what was, in fact, in the supposed to be in the sample. Next. So in this tree, you see those two OTUs where they are located. And next, you will see the other part of the tree and the fact that Stygonema ocellatum was what was supposed to be there. Yes, next. The collector Hans Zimmer was secretary of the Free Association of Botanists from Tyrol. And as you can see in the next, he had published about the cryptogam flora of that place. Next, you can see that he deposited in that year 160 plants in the Smithsonian collection, as you can see in the annual report of the Board of Regents of the Smithsonian Institution in the next figure. And then we can go to the next sample, Druwe 1, which comes from Alaska. So it was taken in 1949, and in the label it was written that it came from Half Moon Tree Reindeer Ranch, and there were geographical coordinates. What did the sequencing give us? It gives us a majority of OTU2 that corresponds to Nostoc, and as you will see, if you click, this Nostoc corresponds to what Druwe has identified in the sample. So next, you will see the tree with all these OTUs, and in the next part of the tree, you will see that indeed it's very related to Nostoc commune sequences. Because we had the geographical coordinates, yes, we can go to the next slide. You can see the landscape of that collecting site, and there are a lot of little lakes. And then concerning the place, in the next slide, you can see that once upon a time there was a reindeer ranch called Half Moon Tree, maintained by the family of Tom Brower, that can be read in the report of the exploration of Naval Petroleum Reserve number 4, which was written by Geological Survey and US Department of Navy. So they explored the region and they made a very informative report. And the next slide shows that they were aware that reindeer were present in these places, but during the period of the explorations, it gradually disappeared. And they speak of this Half Moon Tree ranch. But they say also it's now gone. Yes, the following, by looking a bit about Tom Brower, it was obvious that between 30 and 50, he spent his life in this ranch. And these are the remains of the coral that was taken back, but still some remains are can be photographed in 2007. So it's a kind of testimony of foregone activity in this place. Next one is the collector, George Lano, was a lichenologist who was working in polar regions. He died in 2003. And you can see in the next, if you click, he's very famous for a monograph of the Umbilicariaceae, lichen family in the Western Hemisphere. But also afterwards, he changed career and he became involved, as shown in the next click, in the polar programs at NSF. So everything comes together. So next slide is the next samples. Comes from Antarctica, from a different, the Ross Island, Cape Royds. Again, there were additional information on the labels that were written and are not on the internet, that it was a terrestrial sample and included Nostoc commune and Formidium autumnale. What did the sequencing tell us, in addition to the card of the map of the island? That there was a strong dominance of Microcoleus, and next was Nostoc, OTU 1 and 2. And in fact, this fits together. The next click will show you that. We had nostoc, and you must know that because of taxonomic revision, a lot of Formidium autumnale is now merged into Microcoleus. So it could be that at that time, both were mistaken. So the tree will show that again with Microcoleus and nostoc present. Next is the next sample comes from the same place, the same day, the same person. So they were probably taken close together. And here they speak about Formidium autumnale. And if you look at the sequencing data, indeed, there is a majority of Microcoleus, and as I said, it was probably it would be the new name of what they called in the past Formidium autumnale. So this fits together, and you can see in the tree also all the OTUs present and the Formidium autumnale present also. So in the next slide, I was interested to see that David Nutt is really a very important person. He was a Navy officer, he fought in the war, but he was also a real good scientist. So he was commander in the second US Navy Antarctic expedition that was at the time that those samples were taken, and in his report he's describing the visit to Cape Royds where he took the samples. But afterwards he did a lot of research on his schooner, Blue Dolphin, and he also was, as shown by the next click, involved in the first measurement of composition of ancient air in Greenland ice. So this is a paper by Schollander and et al. In 1956. And in fact, they are the kind of fathers of the modern studies of ice bubbles in ice cores, air bubbles in ice cores. And his team also measured the age of Greenland ice. So this David Nutt died in 2008 after a long life. The next sample is Drouet 17, is the one where we had very few reeds. It comes from Cathedral Crags in Deception Island. And in the next click, you see what the sequences were giving. Even if there were very few reeds, the majority was a Microcoleus, and it was indeed supposed to be a Microcoleus vaginatus, as shown by the next click, as Drouet has identified the sample. So it was, again, as shown in the following tree, a good match with foreseen identification. Next slide is about the collector. And in fact, I could only find one publication with Cressit, and it's probably due to the fact that it might have been a student who came with his brother to the Bishop Museum Antarctic expedition of 59-60. The Bishop Museum is situated in Honolulu, and they did quite a lot of investigation, especially for insects, and the Cressit line is coming from this Cressit researcher. Yes. And next slide is the last sample, which comes from another part from Antarctica in Victoria Land, near Lake Penny. Again, on the labels, there were geographical coordinates that were not on the internet and allowed to show where the place was or about was to be. And it has a lot of diversity, as you can see. And the rivularia, which is supposed to be, at the next click, you will see it, which is supposed to be in the sample, is present, but at a quite low abundance. But maybe that it's also a case of patchiness of the sample, because it is finally it is there, and rivularia tend to make small balls. So it may just be patchiness. Next is, you see the two parts of the tree. And in the second part, you will see that it really fits with Rivularia. So next is the collector, Edmund Schofield. Is also a very interesting person. He was very active with cryptogams in Antarctica, but he was much more than that. As can be seen in the next click, he was a conservationist, director and president of the Toro Society and of the Toro Country Conservation Alliance. He fought for the conservation of Walden. And so he has been very active also with Arctic pollution, and he published a lot. Yes. So next, the last slide is, we can answer something to the two questions about the first one. Next, the click is, yes, we can get useful information, but it's also limited by the information on the label. For example, we saw a lack of geographical coordinates for many samples. And of course, the 16S ribosomal amplicon has some conservation and lengths that could be pose challenges to make real good inferences. The second question, and it was also unexpectedly a meeting with past scientists who worked in polar region and had very interesting lives and contributed very much to polar science and science in general. And how should we preserve our various samples to give a useful picture? The next click will tell you that the air drying that was used by Drouet seems to be a good strategy. But as the next click will see, we need a good and detailed metadata that also should be carefully conserved for data. So it means a data management plan. And then I would like to thank Smithsonian Institution and the FNRS who funded this work. Thank you.
Thank you, Annick, for your talk. And now we have a short break. We will be back in an half hour at 14 in the Greenwich Mean Time.